Good morning, yo. Welcome back to your favorite science podcast. You know me, I'm your host with your co-host, Guillermo Regan. Today we have here two very special guests. The first one is ex-biologist Omar Hernandez. A round of applause for him. To my right, we have here my very good friend and a special guest, Nicolas Aguinaga. Please welcome him.
So guess what, everyone? We're gonna start this episode with a couple of questions from our fans. So first of all, we got this guy from Instituto Thomas Jefferson that doesn't know what the structure of DNA is and how it replicates.
What? This is very important info this guy is missing out on.
Well, what are we waiting for? Let's give these kids the knowledge they need. Omar, are you ready for this?
Wait, you wanna do the thing? Like right now?
Yes, bro, go for it. You should do it.
Well, everyone, let me introduce you to the most advanced scientific software on the line. Welcome to Biocraft. We will use an entire digital world to teach you about what really makes us be us. So, without further ado, welcome to the science of the small.
Hello! To be here, we had to shrink to a size of about 2 nanometers. Right behind me, you can see the foundation of human life, deoxyribonucleic acid, aka DNA. So why are those stacked blocks even important? What are they made of? How do they work as a code? Emiliano, which is right here, and for some quantic reason became a dog, will explain to us how all of this works. Emiliano, please.
Alright, thank you, Memo. The molecule of life, as you can see, is a double helix consisting of two identical strands called polymers. These are made up of single repeating units called monomers, which, in a large model, can be seen every time one strand joins the other. Yes, behind me you can see one of them, but let's take a closer look. A nucleotide, which we use to specifically refer to each one of these monomers, consists of three main parts: a phosphate group, a sugar, and a nitrogenous base.
Now, nitrogenous bases exist in 4 types: adenine and guanine, known as the purine bases, and thymine and cytosine. The pyrimine bases. Adenine always pairs with thymine, while cytosine always pairs with guanine. Now let's go back to our complete double helix structure. Opposite strands match, as you can see, with multiple base pairs. These pairs, as we said, A with T and C with G, these create a sequence unique to every organism, making DNA some form of information storage for the creation of proteins.
Alrighty, now that we know what DNA is, it is time to talk about replication. First, we will answer a question: where and when. DNA replication, or the process of creating new DNA, occurs in different places depending on which type of cell we are talking about. On eukaryotic cells, DNA replication occurs in the nucleus. On prokaryotic cells, on the other hand, it occurs on the cytoplasm. But for now, let's focus on eukaryotic cells. As for when it happens, well, DNA replication happens just before cell division in a stage called interphase.
Excelsior! Thank you, Emiliano. Now, the question, who? In DNA replication, there are 4 enzymes that we can consider to be our main characters. These enzymes are helicase, primase, DNA polymerase, and ligase. Let's check them all out as we explain how replication takes place. Let's take a look at this model.
And well, the process begins with specific spots in the DNA strand that have been identified because of their sequence. These are called origins of replication. Helicase is known by his bodies as the unzipping enzyme and depicted as previously shown with the purple blocks. So why that nickname? You could ask. Well, it means that helicase unzips or breaks apart the two strands of DNA that compose the double helix structure, creating a replication fork. Helicase does this by breaking the hydrogen bonds that hold them together.
Next, we have DNA polymerase. At work, DNA polymerase is liked and appreciated by everyone. The closest enzymes to him have even nicknamed him the Xerox. Yes, like the printer. The function of DNA polymerase is essentially to synthesize new base pairs by taking each strand as a template. Since each strand has a particular base for every nucleotide, and since the rule is that each base has a specific pair match, polymerase can weld a new nucleotide for each individual base. When this enzyme is done, there will be two identical copies of the original double helix. Pretty cool, huh?
Third in line we have primers, or otherwise known in the office as the manager. And it only manages DNA polymerase by the way. The function of primase is to create RNA primers, which not only tell DNA polymerase where to start adding nucleotides, but also initiates the process by adding a small amount of nucleic acid.
And finally, and definitely not least, ligase, better known as the glue, and no, it is not called like that because it holds friendship together. Ligase function is to glue together two DNA fragments with loose ends during replication. In what could probably be Bill Nye's words, ligase catalyzes the formation of phosphodiester bonds needed for DNA replication. Finally, in regards of enzymes, let's not forget that DNA polymerase runs in the 5 to 3 direction towards the fork. What this means is that in the leading strand, which runs in the same direction, is synthesized continuously, but on the lagging strand, as it runs in the opposite direction, is synthesized in fragments.
As a conclusion, we now know that DNA replication is a semi-conservative process. Each strand in the double helix acts as a template for synthesis and a new complementary strand. We hope to have clear at least the most important fact about DNA.
Well, ladies and gentlemen, I'm afraid we have reached the end of this episode. Thanks for joining us in this very educational topic. I'd like to thank our guests for being here tonight and helping us out with this topic, okay? Anything you want to add?
Remember to use mask because we're still on a pandemic.
Yeah, great tip, man.
Thank you so much for the invite.
No, thank you for being here, man.
Have a great day.